Hello everyone, welcome to Bioinformatics. Today we will be looking at what are biological databases. In this video, we will be understanding about the introduction to biological databases, classification, types, features, and some examples of biological databases. So what is a biological database? In simple words, we can define it as a collection of data which could be structures, searchable, updated periodically, and can be cross-referenced. It stores biological data in the form of electronic information. Now, why do we need biological database? The purpose of biological databases could be for systematization of database, availability of biological database, and analysis of computed biological databases. Now, what are the features of biological database? It could be heterogeneity, which means varied types of data, high volume data, which means massive content, uncertainty, it means deviation from the original data, data curation, that is the selection and organization of data, then data integration, that is it combines data. Next, data sharing, which means to provide access to all the users. And finally, data dynamics, that is the data management. The different types of data which could be classified under biological databases are genome database, sequence database, structure, microarray, chemical, pathway, enzyme, diseased, or literature database. Now, under the types of biological databases, we could classify as primary database and secondary databases. So what is primary database? It contains biomolecular data in its original form. The experimental results are submitted directly into the database by the researchers, and the data are essentially archived in nature. Once a database accession number is given to a kind of data in the primary database, then that data cannot be changed thereafter. Some of the examples of primary database are gene bank, EMBL, DDBJ for DNA and RNA sequences, and for protein sequences we have SwissProt and PIR. We will look into each one of these databases in detail. Beginning with gene bank. It is a database associated with NCBI and it includes sequences from publicly available resources. This gene bank is a genetic sequence database and annotated collection of all publicly available DNA sequences. It forms a part of the International Nucleotide Sequence Database collaboration. Now in this slide, the second half of the slide is what the home page of gene bank would look like. Next primary database is EMBL. EMBL is an abbreviation which stands for European Molecular Biology Laboratory. It is again a nucleic acid database that falls under the umbrella of EBI. EBI is European Bioinformatics Institute. It was established in collaboration with the DDBJ and Gene Bank. And EBI's sequence retrieval system is again a huge network browser for data banks in molecular biology, integrating and linking the main nucleotide and the protein databases worldwide. The third primary database is DDBJ. It is an abbreviation which stands for DNA Data Bank of Japan, and it is a biological database that collects the DNA sequences. It is located at the National Institute of Genetics in the Shizuoka Prefecture of Japan. It is also a member of the International Nucleotide Sequence Database Collaboration, or INSDC. And below this, what you see is the home page of DDBJ. For proteins, as we just now mentioned, the two major classes of databases are SwissProt and PIR. SwissProt is a curated protein sequence database which strives to provide a high level of annotation, such as describing the functions of a protein, the domain structure, the post-translational modifications, variants, etc. Swissprot was created in 1986 by Amos Weirach and Swiss Institute of Bioinformatics, or SIB. Below in this slide, what you see is the home page of Swissprot. The next primary database exclusively for proteins is PIR. PIR is an abbreviation for Protein Information Resources, which is an integrated public resource for protein information which supports all the genomic and proteomic resources based on scientific discoveries. This PIR maintains the Protein Sequence Database, which is an annotated protein database containing over 283,000 sequences covering the entire taxonomic range. Now we move on to secondary databases. It contains data retrieved from the results of the primary database. This data in secondary database is either manually created or generated automatically. It contains some valuable information such as about mutations or evolutionary relationships. We will look into a few examples of secondary databases like ProCite, ProFab, etc. ProCite is a protein database. It consists of entries describing the protein families, domains of functional sites, as well as amino acid patterns and profiles in them. They are manually curated by a team of Swiss Institute of Bioinformatics and tightly integrated into Swiss protein annotations. The second example of secondary database is PFAM, or Protein Family, which includes the annotations and multiple alignments generated using hidden barcode models. The general purpose of PFAM database is used to provide a complete and accurate classification of protein families and domains. Under structural databases, we could classify PDB, SNP, and CAD. PDB is Protein Data Bank, which comprises of PDB of Europe and PDB of Japan. PDB of Europe is abbreviated as PDB-E, and the PDB of Japan is abbreviated as PDB-J. The second structural database is SCOP, which is Structural Classification of Proteins, and the third is CAD, which stands for Class, Architecture, Topology, and Homology of the proteins. They all contain information which is generated from X-ray crystallographic studies or from NMR experiments. Apart from primary secondary databases, we also have something called as specialized databases. These databases are exclusively meant for certain type of organisms or certain type of study, or it could be even to classify an exclusive type of disease which could be related either to human beings or to plants or to animals. For instance, to study the model organism, there could be a database. To study only the rice species, we have a database. To study only the microorganisms, we have a database. To study only the inherited disorders or genetic disorders in human beings, there is yet another database. So when a specific database caters to an exclusive need, we call it as a specialized database. There are hundreds of specialized databases exclusively meant to cater to special needs. Some of the specialized databases are shown in the current slide. For example, N.G.P, which means Human Gene Mutation Database. So all the gene mutations pertaining to the human beings can be obtained from N.G.P. Next is called as OMIM, that is OMIM, which stands for Online mutation inheritance in man, or it is also called as online Mendelian inheritance in man, which means all the inherited disorders pertaining to mankind can be studied from OMIM database. You can obtain the whole sequence of the gene which is responsible for a particular disease. For example, if you want to study about, let's say, a disease like Duchenne muscular dystrophy or DMD, then the gene which is responsible for Duchenne muscular dystrophy is the dystrophin gene. You could probably get the whole sequence of the dystrophin gene present in a Duchenne muscular dystrophy patient. Or we can give the timelines of curing or the methods of curing or the different databases or organizations which could help in treating a Duchenne muscular dystrophy, or what are the diagnostics necessary, and so on. All kinds of information relating to an individual inherited disease pertaining to mankind can be obtained from OMIL. The next specialized database is TransFate. It stands for Transcription Factors Database. It is exclusively meant for the eukaryotic transcription factors and not prokaryotics. So if you are looking for the study of any eukaryotic organisms, you could log into TransFate. And finally, we have the R-fam, just like how we had the P-fam in the subsequent few slides before. We saw P-fam, which is a database meant for protein families. Similarly, we have an R-fam here, which stands for RNA family databases. So all the information pertaining to the RNA can be obtained from R-fam. So that is regarding the different types of databases. Now there is another classification based on data access. So how easily can you access this biological database? Is it paid, or is it free? Can you use it or can you browse it? Now, for example, that can be classified into 6 different types. The first one is publicly available, whereby anybody can log in and download the information. The other one is it is available only with a copyright. Then the third one is browsing only, accessible, but you cannot download. You can browse, you can read through, you can work on it, but you cannot download the particular content. Academic but not freely available. Some of the academic institutions would have already signed up a lease for so many years, and they would have paid a huge amount of money, and therefore those academic institutions will be given a login ID through which the students can access freely. The student need not pay. The academic institution would have already paid for that particular database. And proprietary, commercial, and then restricted. Restricted, however you try, you cannot enter. Only on payment of some amount you will be able to make use of the database. Thank you very much.